All right, thanks everyone for joining. And just to introduce myself, my name is Bill. I'm the lead for the GDC user Services team, and today I would like to talk to you about GDC BAM slicing. Okay, so just to go over a very quick agenda, we're first going to talk about an introduction to BAM slicing, what that is and what this feature is in the GDC. Then we're going to do a quick demo on BAM slicing from the data portal, some instructions on how to perform BAM slicing from the GDC API. Then we're going to go over some tips and troubleshooting for when users BAM slice. And then finally, if we have any questions from participants, feel free to type that into the chat window as the webinar goes on. And at the end, we will answer your questions. And of course, any questions that you don't think of now or you come up with later, you can always send to the GDC help desk, which will show the email at the end of this presentation. So starting with an introduction, just to talk about BAMs and what we have in the GDC, the sequence read alignments that are in the GDC are stored in binary alignment map format, or BAM, which I will be calling it for the rest of this presentation. As most people who've worked in bioinformatics know, BAM files can be very large in size due to their genomic scale. And the entire genome may not be required for your study. If you look in the GDC, for whole genome sequencing alignments, the mean size of them is 164 gigabytes, whereas the median is 103 gigabytes. So these can be very, very large files. And we are aware that, many groups don't have large storage spaces available. And just to kind of show an example here, this is a query for the TCGA LUAD project, which is lung cancer. And if one was to download all of the whole exome sequencing BAM files from that one project, this would be almost 2 terabytes, which can be pretty big, especially for a single personal computer. So to solve this problem, the GDC has a feature called BAM slicing. This helps users decrease the file size, especially if they need only a part of the genome. So this allows for a certain region, set of regions, or a certain region to be downloaded from the GDC in BAM format. So you go from large BAM to smaller BAM. This region can be things like entire chromosomes. They can be specific ranges within chromosomes, and also can pull out specific genes if the exact range isn't known. The slicing is available in a couple of ways. The first being through the API, and the second being the data portal. And we're going to show you just a little bit about both of those. So to compare both of the methods that I just mentioned before, the portal and the API, both can use genomic coordinates to specify the region. If you know the genomic coordinates, either of these methods can be used. However, if you want to use gene symbols, such as like KRAS, to specify the region, that feature is only available in the API. The number of BAM files also matters and which method you might want to use. So if you're using a small number of BAM files, if you're downloading from a small number of BAM files, both of the methods can be used. And I'm going to show you why that is in a second. But a large number of BAM files, we would recommend using the API due to its automatable features. If you are accessing it programmatically using such as a Python script or something like that, of course, that would be only available through the API. But the portal does have a user interface for those who are who would like to use something like that. And just to make it clear, both of these methods do require dbGaP access to whichever project the BAM is in. So I want to start out just with a few caveats, and just as I said in my last point before, and I can't stress this enough, that all of the BAM files in the GDC are controlled access. It, in the same way, all slices of BAM files are controlled access. So make sure if you are BAM slicing, the that you keep them secure in the same way you would regular BAM files and abide by all dbGaP regulations. Um, The GDC has an RNA-seq pipeline that generates three BAMs: a genome, a transcriptome, and a chimeric. Because the transcriptome BAM isn't sorted in the same way as the other two, BAM slicing cannot be performed, and that's because they don't have a BAM index file. Speaking of not having a BAM index file, occasionally the BAM files may be missing their index file. These will need to be downloaded in full before they can be sliced. And if you ever do come across these, we keep an eye out for them internally, but if you happen to find one, always feel free to contact the GDC help desk to let us know. That would be very much appreciated. So, to start out, we're going to talk about BAM slicing using the data portal, and this is going to be a small demo. So I'm going to open up Google Chrome here, and we're going to start out at the GDC data portal. I'm going to go to the repository, which is where the BAMs live. And here we have a big list of 931,000 files. So this is BAM slicing. Of course, we want to use a BAM. So I'm going to go down, looking at the filters on the left to data format, and I'm going to filter by BAM. So only the files that are in BAM format are showing now. I'll choose the first one, which is a whole exome sequencing BAM from the TCGA BRCA project. So in order to start the BAM slicing process, it's very easy. There's just a button at the top right that says BAM slicing. Once you click on that and you're logged in, you will see this wizard come up. If you're not logged in, a notification for you to log in will come up. So in this field is where you would type the coordinates, and this is the UI, so coordinates are required. As it says in the bottom here, alternatively, you can enter unmapped, which will provide the unmapped reads. And as you may or may not know, the GDC keeps all of the unmapped reads in the BAMs, in the alignments, so that the original raw data can be recovered from the BAM file. So starting out, I'm going to put the first line, CHR2 colon, and then 1-20,000. So what this is saying here is we have chromosome 2, and the first through the 20,000th base, I would like to be in this slice. For the next line, I'll enter CHR19 with nothing after it. This indicates that I would like to download the entirety of chromosome 19. After I'm done here, I will click download, and this will bring up a preparation window, and you should see starting in the bottom left, a download will start. So give that a few seconds. These can be pretty big. And what I'm going to do is cancel this so that it doesn't interrupt the presentation later. So going back to the presentation, next, I would like to tell you about BAM slicing using the API. And this is done in a command line interface. If you have other methods of using an API, this can be done using those, I'm sure, like through Python or through perhaps a more user interface method for interfacing with an API. But all the examples here, we're going to use curl. So as I said before, this is the recommended method for downloading slices from many, many BAMs. It really depends on how much manual work you're willing to do through the user interface. This, of course, can be automated with something like batch scripting or Python or another programming language. And another good feature of the GDC API is that it allows for the GenCode 36 gene symbols to be specified. So these are the gene symbols that are more or less human readable, like KRAS or BRCA. These aren't the ensemble IDs with the long number. So at the bottom here is what one of the API calls will look like. And we're going to get a little deeper into that right here. So this is a curl command, and the blue texts are comments that just give a little more information on what we're looking at for each. So starting out with curl, this is going to be a git command. And if you just run curl and you don't specify the -x parameter, it defaults to git. So we're going to start out with a header, and this header is going to be the token. Like I said before, you do need dbGaP access, so you will need to pass a token to the API. And in this case, the token is stored in a variable named token or whatever you want to name it. So next, the GDC API URL is specified, followed by /slicing/view/ and then whatever the UUID for the file is. So this would be the file ID. So after the file ID comes a question mark, and the regions are specified here. So we would have regions equals chromosome 1 colon the start dash end and then ampersand and then the next regions, and then after that you could do something like GenCode equals, and then that's how the gene symbol is specified. Then lastly, once the URL is done, you would specify the dash output parameter, which just gives you the name of whatever you want the name of the BAM slice to be. So you could use a unique ID, you could use something to do with your regions, whatever you'd like. The post method, we recommend using this if you're gathering a lot of regions, because there is a limit to how big the URL can get. Additionally, if this allows you to keep things better organized for your specific project, feel free to use this too. So the only differences here are first, you would specify that this is a post command. Token is the same. Another header is added that specifies that the data that you'll be posting is in JSON format. So it's content type application/JSON. After the header and the UUID, and the UUID is the same except it won't have the parameters at the end. You would pass the data parameter, and this is the payload file. And this is basically the same thing as what was shown in the git command, except that it is specified as a JSON file with the keys on the first level being the regions or the GenCode, and the values being a list of the regions or the GenCode symbols. So that payload file would be posted, and of course the output file is the same as before. So as an example, here is an API call, and in this case, we have this call will download bases 1 through 20,000 for chromosome 2, the entirety of chromosome 19, and then the KRAS gene on chromosome 12. And I do want to specify that right here, I left out a region equals. So that should be between the ampersand and the chromosome 19. Usually we would do live demos, but because of the sensitivity of the data, we are not doing that here. So lastly, I want to talk about some BAM slicing tips and troubleshooting. So the first thing I want to tell you is that GDC BAM slicing does not return multiple copies of data when the same region is requested multiple times. So KRAS is a gene on chromosome 12. And if you were to query for chromosome 12 and the KRAS gene, they would be redundant because KRAS falls within the chromosome 12 region. So theoretically, this should return the same result. And as you can see, I ran this before, KRAS and chromosome 12.bam and chromosome 12.bam, and they have the same exact value as a demonstration. The If no region or gene is specified, this will return only the BAM header. And the BAM, this might be useful if you want to look at the BAM header without downloading any piece of the BAM. Additionally, if you think you specified a region and you only get a BAM header, it's possible it's empty or it's just something wrong with the region parameters. So that's a good way to troubleshoot if you're only getting a header. And then if you request an empty region, um, you will not get an error, and you won't get an error saying this region has no reads. You'll just get an empty BAM with just a header and no reads. So another thing I'd like to let you know about is that if reads overlap the beginning or the end of the region of interest, so if you query for 10,000 through 20,000 and only half of the read is within 10,000, like it starts at um, like 9,950 or something, it will the entire read will be provided. Also, reads that do not fall within the region of interest but are paired with reads that do will not be provided. So you will only get one of the pairs of the reads and not the other. If they both fall in the region, you'll get both pairs. Additionally, no BAI or BAM index files are created during BAM slicing, and users must create their own. So if you're using Linux or Mac, an easy way to do this is using SAM tools. I know there are other other pieces of software that can do this, but an easy way to do this is just do SAM tools, index, and then whatever your BAM slice is named. This will create an index file that just appends .bai to the end of your BAM. If you use Windows, an easy way to do this is to use IGV, which is a software package developed by the Broad. And I'm going to demo this real quick. It's very easy. So here, if you look at my terminal, I have a BAM file called gene.bam. So if I go to IGV, I click at the top here at the tools in the toolbar, and I click Run IGV Tools. This will bring up a prompt, and normally the genome would be human, but I changed it to banana just to be safe. And for command, you would click on Index. For input file, you would browse and go to whichever BAM file you were looking for. In this case, this is the one I showed you before, gene.bam. I'll select that and then click Run. The message says done at the bottom. And if I go back to the terminal and check the directory again, I will have gene.bam.bai. The BAM index file is here. So going back to the presentation, we're going to talk a little bit about troubleshooting before the end. So if you're not sure that a BAM slicing call worked, and perhaps you don't want to pull up the actual sequences just for security or because perhaps you're looking at many of them, an easy way to do this is to look and run file and then whatever the name of the BAM is. And if you wanted to look at like all your BAMs or something, you would just do star.bam. And this is an example of what it is. And the top three here are legitimate, valid BAM files. And the bottom one, which is one that I purposely had fail, is JSON data. And this actually makes sense because GDC provides errors in JSON format. So if you ever see JSON data when you run this command, that means that an error is there. And if you wanted to look at that error, you could use that BAM and open it into a text editor. It'll open because it's just JSON data and not actual um, binary data. If you actually want to look at a BAM that you've sliced and you don't mind the sequence being shown on your screen, and I just want to remind you always be careful, especially for dbGaP protected data, which is all BAMs from the GDC. You would type samtools view and the slice.bam. Now, before I was talking about GenCode symbols, and you may not know, like, what is the GenCode symbol for the gene that I'm interested in? And you might be interested in using them. A good way to do this is you can go to the gdc.cancer.gov exploration page, and you can query for genes there, and there's a big list of the genes that have the greatest number of mutations. That might be helpful, especially if you type in the autocomplete. And then additionally, you can go to gdc.cancer.gov, and just to kind of show you where that is, if you go to gdc.cancer.gov, you go to About the Data, down to GDC reference files, and if you scroll down under annotation files, you will see the GenCode v36 GTF file, and this has all of the gene symbols in it. So you can download that and look, or you could use coordinates too. That's always an option. Okay. That is the end. So we'll take any questions from participants, and just to let everyone know, here are some useful links. And if any questions aren't answered here, or if you have any questions about BAM slicing or anything GDC related in the future, we recommend you contact the GDC help desk at this address right here. This leads directly to my team, and we will answer any of your queries in one business day. Thank you so much, and happy to take any questions. Yeah, I don't see any questions yet, so I'll just give it a minute or so. Okay, well, it doesn't seem like we have any questions at this point. So, yeah, thank you so much for joining. And yeah, I hope you have a good rest of your day. Thank you.
Thanks, everybody.